In this class, I'll discuss about LDL metabolism. LDL is called as a low density lipoprotein, which is a major carrier of cholesterol in blood. And this LDL molecule is made up of an outer shell. This outer shell will have phospholipids and unesterified cholesterol, and they will have a protein called as ApoB100, and this ApoB100 is recognized by target cells. And this LDL molecule, this outer shell, will cover an hydrophobic core. This hydrophobic core is made up of will contain cholesterol, and this cholesterol will combine with a fatty acid. That means this cholesterol is an esterified, and this fatty acid is linoleate, and this is a polyunsaturated fatty acid. And other than liver and intestine, all cells will obtain cholesterol from plasma with the help of LDL molecule. They will not synthesize cholesterol on their own. There won't be de novo. Maximally, they depend on obtaining cholesterol through an LDL molecule rather than de novo synthesis. So the primary source of cholesterol for these cells is LDL. And the process through which cells taking up of cholesterol through LDL molecule is called as receptor mediated endocytosis. So how this will take, we'll see. This is a cell which is having a pit, and this pit will contain receptors called as clathrin receptors. This is a coated pit, and this pit is coated with the clathrin receptors. Okay, this cholesterol will contain ApoB100, and this ApoB100 is recognized by these clathrin receptors. So whenever LDL molecule comes to the cell, this ApoB100 will bind with the clathrin receptors, and they will form an LDL clathrin complex. And this complex is internalized. Along with the LDL molecule. So these are clathrin receptors, and these clathrin receptors will be having LDL molecule with a ApoB100. So there will be an internalization of this cell membrane, and they will form an endosome. They will form an endosome. This is endosome. Endosome. So after formation of endosome, what will happen? This membrane will have proton pumps. So these membranes will have proton pumps, and these proton pumps will cause pumping of H plus ions into the endosome, and they will increase, they will cause acidification, acidification of endosome, acidification of endosome. This acidification of endosome causes separation of a receptor and LDL molecule. What will happen? This clathrin coated receptors are separated. These clathrin coated receptors are separated, and an endosome with LDL molecules will be there. LDL molecules will be there. And this clathrin coated receptors will be recycled back to the cell membrane, and they will form a clathrin coated pits once again. So these are clathrin receptors are recycled back. And now we'll see the fate of this LDL molecules. Now this this endosome will fuse with the lysosome. Lysosome is there. This is a lysosome, and this endosome and lysosomes will form fuse, and they will form endolysosome. Endolysosome is formed. This is an endolysosome. And this lysosome will contain, they will contain proteolytic enzymes, and these proteolytic enzymes will break the protein part into free amino acids, and this lysosome will also have lysosomal acid lipase, and this lysosomal acid lipase will cleave the cholesterol ester into free cholesterol plus fatty acid. So this lysosomal acid lipase will act on the cholesterol ester which is present in the LDL molecule, and they will free the cholesterol and fatty acid. Sometimes this clathrin receptors, they are not recycled back. Sometimes they will join with the LDL molecules, and they will fuse with the lysosomes, and they are degraded. So not complete recycling of clathrin coated pits will occur, but some of the receptors will be degraded along with the LDL molecule. Now we will see the what is the fate of this free cholesterol. This free cholesterol, or it is called as unesterified cholesterol. What is the fate of this? So when this cholesterol, they can move to the membrane, and whenever this cholesterol moves to the membrane, they help in the fluidity, maintenance of the fluidity of cell membrane. Fluidity of cell membrane is maintained by the cholesterol. It acts as a buffer in maintenance of fluidity of cell membrane. And whenever this cholesterol reaches skin, they will form vitamin D. And when cholesterol reaches adrenal glands, they will form steroid hormones. And cholesterol will help in formation of bile salts and acids. Acids. Now, this is the fate of the free cholesterol. Whenever cholesterol is more, this free cholesterol is again re-esterified. So, remaining cholesterol will be re-esterified. Re-esterified. So, why this re-esterification has to occur? It helps in storage of cholesterol. Storage. This is stored inside the cell. Now, this free cholesterol, what it will do for esterification? This free cholesterol will activate an enzyme called as ACAT. ACAT. What is this ACAT? This ACAT is acyl CoA cholesterol acyl transferase. Acyl transferase. And this enzyme will cause re-esterification of free cholesterol and causes storage of cholesterol. And in the starting, we have seen that inside this LDL molecule, cholesterol ester is there. Cholesterol, which is combined with the fatty acid, and this fatty acid is a linoleate. But in this re-esterification inside the cell, this cholesterol will have a fatty acid which is made up of either it is made up of oleate or it is made up of palmitoleate. These are the fatty acids which are attached to the free cholesterol after degrading inside the cell. When LDL molecule is transporting cholesterol from liver to the cells, the fatty acid which is attached to the cholesterol is linoleate, but inside the cell, again re-esterification, the fatty acids which are used are oleate and palmitoleate. So this is the basic mechanism which is involved in absorption of cholesterol through LDL molecule other than liver and intestinal cells. Now we will see the regulation of LDL LDL molecule. How this is regulated through receptor mediated mechanism. Regulation of LDL metabolism. This is a cell and LDL molecule is coming here, and there is an influx of cholesterol. Influx of cholesterol. This influx of cholesterol into the cell causes inhibition of transcription genes which are encoding HMG CoA synthase, HMG CoA reductase. These are the main enzymes which are involved in the synthesis of cholesterol. These influx of cholesterol will causes inhibition of transcription genes which are encoding HMG CoA synthase, HMG CoA reductase, so that these enzymes are not synthesized and de novo cholesterol synthesis doesn't occur. And LDL receptor synthesis is regulated via SREBP pathway. This SREB pathway is a regulatory step for cholesterol synthesis. Through the same mechanism, this LDL receptor synthesis is regulated by SREBP pathway. And there is one more mechanism which is a recent advance which is seen in LDL metabolism, where there is a protein molecule that is called as a proprotein convertase subtilisin or kexin type 9. This is called as PCSK9. This is called as a PCSK9. What it will do, it will regulate the recycling of the receptor to the cell surface. This is how LDL metabolism is regulated. Now this ends the LDL metabolism. Thank you.